Our next speaker is Dr. Bastian Bentlage. Bastian is an associate professor of bioinformatics at the University of Guam in the Western Tropical Pacific. At the University of Guam, Dr. Bentlage's research focuses on the impacts of environmental degradation and climate change on corals and their symbiotic algae and bacteria. Please join me in welcoming Dr. Bentlage.
Okay, half a day, good morning. As was said, my name is Bastian Bentlage, and I'm an associate professor at the University of Guam, more specifically at the Marine Laboratory in Guam. And my talk today will focus on the impact of sedimentation on our nearshore coral reefs. And before I start, I would like to orient you, where is Guam actually? Guam is a US territory in the tropical Western Pacific, and being near the equator is surrounded with coral reefs. And those coral reefs support local fisheries and associated cultural practices that are thousands of years old, and are an attraction to our tourism industry, which is the biggest industry on island. We're all familiar with the fact that climate change and rising sea surface temperatures impact coral reefs, lead to the breakdown of corals with the endosymbiotic algae, which ultimately leads to coral bleaching and death of corals. This is something I don't actually want to talk about today. I want to talk about the impact we have more directly on coral reefs through coastal development and land management practices. This excavation you see here is an example of a housing development near the Marine Laboratory. And we have a pronounced wet season in Guam, so once it started raining, land or the excavated soils, exposed soils weren't properly contained, washed out into the ocean and smothered our nearshore coral reef and killed a lot of coral in the process. And in southern Guam, we have this phenomenon where we have the formation of a lot of badlands, exposed soils where nothing grows anymore. The soils are moving, seeds can't properly germinate, and they're a result mostly of wildland arson. Hunters will set the savanna grasslands on fire because the new shoots attract deer and support or make hunting easier for deer and pigs. But the problem is now we have these exposed, degraded soils in which nothing grows anymore. And those badlands are co-located with our watersheds, with the rivers in Guam that are largely restricted to southern Guam. So during the wet season, when it rains, large amounts of soils are transported by the rivers into our nearshore reef ecosystems. And I would like to introduce you to one of our model sites for studying the effects that runoff has, Fuha Bay. Fuha Bay is in the Lasafua watershed, and there's a couple of different terrestrial habitats throughout that watershed, mainly savanna grasslands, forests, but I also tried to indicate the badlands on the ridges, the source of sedimentation. Not too long ago, or actually 50 years ago, but in the last century, up to the early 1980s, Fuha Bay was home to about 155 coral species, so a fairly diverse reef. But that diversity has declined by half, down to now 84 coral species that are recorded, largely due to sedimentation. And what I want to point out is that sedimentation in Fuha Bay and really all of our southern bays in Guam is common, but flushing, the removal of sediments is pretty rare, and the reason being waves swells will have to have the right direction so that waves can actually remove the sediment. If we zoom into Fuha Bay, there are about 1,700 tons annually of sediment that are deposited. But those sediment deposits or the sedimentation is, as you may expect, not uniform. Sedimentation is severe or very severe near the shore where the river meets the ocean, and then declines to light or non-existent anymore the further away we get from shore. And consistent or coinciding with that, coral cover, so like the amount of bottom covered in coral, increases the further away we get from shore. The size of individual coral colonies increases, as the species diversity and richness, as you may expect. So what are the impacts of sedimentation on corals, actually? Corals have to invest energy, so there's an energetic cost to being smothered in sediments that settle on coral surfaces. Corals use mucus that they have to produce to bind the sediments and then shed them. Ciliar motion may remove sediments. And that energetic cost, as you may imagine, increases the susceptibility of corals to disease. Sediments may bring pathogens into the ecosystem, so all that may also increase the prevalence of disease, coral diseases. The other thing that happens is sediment blocks sunlight. I mentioned that corals have the symbiotic association with algae that live in their tissues, so reduced sunlight means reduced photosynthesis, which again has an energetic cost. The other thing that happens is, so now we have our susceptible corals that may die due to disease or starvation, but we also have less recruitment because larvae cannot settle in soft sediment, on soft sediment bottom. They need exposed hard bottom substrates, so sedimentation also reduces the amount of recruitment on our reefs. This is a figure that summarizes Fuha Bay in particular. The curved line represents coral diversity in Fuha Bay. On the left, we have no sedimentation. On the far right, near shore, we have very severe sedimentation. And what I want to point out is that essentially coral diversity, the number of coral species, exponentially declines as sedimentation increases. There are a few species that persist in severe or very severe sedimentation conditions, and one of them is Porites lobata, our study organism. On the left, we have a Porites lobata colony growing under moderate sedimentation. You can see the tissues, the colonies intact. On the right, we can see what severe sedimentation does. There's few patches of living tissue left, and a lot of tissue died under the sediment that's covering the coral and led to necrosis. We wanted to know, or one of my students actually wanted to know, what happens in the cells in these corals that are exposed to sedimentation. And she sampled corals growing in the inner bay, near the river, and then further out, so under severe sedimentation and under light sedimentation. And without going into too many details, here in this figure, in this heat map, every column represents a coral sample. On the left, we have the coral itself. On the right, we have the endosymbions, the algal symbions of the coral. And every row represents a gene and its gene expression value. How abundant is that gene in the tissue? Blue meaning low abundance, red high abundance. And the take-home here is basically that both in the symbiont and in the coral itself, gene expression patterns are consistent with the origin of the coral. So nearshore corals and their symbionts have similar gene expression patterns that are different from farshore corals, and we associate that to the chronic stress that they're exposed to. And when we looked more closely into how many genes are actually differently expressed, we find between 1,500 and 1,700 genes. And those were mostly related to energy metabolism and immune response, as we may expect, so like the genes that are needed to cope with that stress. And the data that we collected in the field are consistent with what others have found in their studies in the lab. In addition to looking at the coral and its algal endosymbionts, we also started looking into the bacterial microbiome of our corals in Fuha Bay. So similar to all other living beings, corals have associated bacteria that are integral to their metabolism, and we used DNA metabarcoding to identify the different bacterial species, bacterial groups that are associated with corals in Fuha Bay. We sampled, again, from the inner bay and the outer bay, and during the wet season, the blue plots, the blue bars here, and the dry season, the red bars. The take-home was that in the inner bay, the bacterial microbiome of Porites lobata is more diverse in general than it is in the outer bay. So severe sedimentation, microbial diversity increased, and during the wet season, we saw in the inner bay at least that the bacterial microbiome was more diverse than it was during the dry season, which makes some sense. There's likely bacterial influx, additional bacterial taxa that are transported by sediments through the river into the bay. If we look at the taxonomic composition, what types of bacterial groups can we find? Again, inner bay, outer bay, dry season, wet season. Here, every column is a sample that we collected, a specimen from the field, and every row represents a bacterial group. I want to highlight a couple of particularly important groups, or something that stuck out to us right away. In yellow, yellow represents high abundance, blue represents low abundance. In the Zircomonaceae, a group of bacteria that is, or has been described as bacterial endosymbionts of corals that seem to be engaged in some nutritional symbiosis, are dominant across all samples, across all sites and time points. Then we find also spirochetes, which are abundant, but only in the inner bay. So it's quite interesting. We find few spirochetes in the outer bay, a lot of spirochetes in the inner bay under severe sedimentation in spirochetes. There's a lot of diseases that fall into this group. So what could their impact be? Another group I want to highlight that is more abundant in the inner bay compared to the outer bay are actinomycetes. Actinomycetes have been described as constituent members of coral microbiomes, but they're also extremely abundant in soils. And sedimentation essentially represents soils that are washed into the ocean. We did some statistical analyses, and so what we saw from the heat map seems to bear out here as well, that actinomycetes are significantly more abundant in coral microbiomes in the inner bay compared to the outer bay. And another group that showed up were dinococci, both groups that are abundant in soils and especially abundant in degraded soils, such as we may find in the upstream badlands, or the badlands upstream of Fuha Bay. So this actually then led to the expansion of my research program, or the research program in my lab, where we now began sampling soils throughout the watershed, throughout different habitats. Here, one of my students, Anna Magliari, and I, in the Badlands collecting soils. And one of the things we're particularly interested in is understanding what types of bacteria can we find in degraded soils, and also are these degraded soils selecting for antimicrobial resistance genes. So there's this idea out there, or evidence out there, other studies out there that have shown that antimicrobial resistance genes proliferate in degraded soils. When resources are scarce, bacteria may compete with each other for those scarce resources. How do they do this? With antibiotics, trying to kill each other. But if you produce an antibiotic as a bacterium, you need the resistance gene to not affect your own population. And actinomycetes that I pointed out earlier as being particularly abundant in the inner bay of Fuha Bay, also known to produce the majority of antibiotics and antibiotic resistance genes. So we're now using, and this is ongoing research or emerging research, we were recently funded to use metagenome sequencing to sequence soils, sediments, coral microbiomes to start screening them for mobile genetic elements that may harbor these antibiotics, antibiotic resistance genes, since mobile genetic elements are essentially one way for bacterial populations to quickly spread these traits across populations. And if you think about it, sort of like the proliferation or potential proliferation of resistance genes in the environment pose some ecosystem health risk and probably some, or potentially some public health risks as well. This work, we're also collaborating here with the Guam Restoration of Watersheds, or GROW initiative. And their goal is the restoration of these terrestrial ecosystems by planting trees in badlands to stabilize soils and ultimately reduce or ideally completely mitigate sedimentation in Guam's watersheds. And they're very interested in our work as well, because my hope would be that understanding soil microbial diversity in our watersheds will also help them inform and adjust their management or restoration efforts. And ultimately, this is a Ridge to Reef project where conservation management of our nearshore coral reefs relies also on restoration of the terrestrial ecosystems, because they're all connected. and what we do in one ecosystem may impact another one. With this, I would like to acknowledge all the people who are collaborating or working with me on this. In particular, my lab, the graduate students, master's students, and undergraduate researchers. Without them, this would not be possible. And over the last couple of years, for this project in particular, collaborations at Boston University and Cal State San Marcos. Thank you, and I'm ready for your questions.
Questions for Bastian? Please come down to the microphone.
Thanks very much for that talk. I'm curious, with the increase in the sedimentation, there's a correlated increase in the freshwater runoff, and therefore is the salinity of the bay decreasing as well?
I'm glad you're actually asking this question. We just got the question back from one of our reviewers on this paper. Yes, but to be honest, so we deployed salinity loggers right next to the coral. Even though they're only growing in 3-4 feet of water, the salinity there doesn't actually seem to drop very much. And when you're out in the field, every time it rains, you can actually feel most of the fresh water is just really sitting on top. The drop in salinity really that we measured is maybe 1 or 2 points, not a lot. So it may impose some stress, but probably not the majority, if that makes sense. And so yeah, like the sedimentation, the suspended sediments, they seem to be floating on top of the salt water and then slowly settle down.
Okay, so then is there any change, I must ask, in the composition of the fish fauna there?
Yes, as in it's almost absent. What do you call it? What has become very obvious, now I've lived in Guam for the last 8 years, when the corals are dying or disappearing, we see that during bleaching periods as well, fish will immediately disappear. Without a living, thriving coral reef, the fish will essentially disappear.
Okay, thanks very much.
Thank you.
That kind of leads into my question, actually. But I wanted to play devil's advocate here and see what you say to people when you say, okay, we had 155 species 50 years ago. Now we have 84. People might say, well, you still have 84 species, right?
Mm-hmm.
So, like, what's the problem? What do you say in response to that?
That's a good question. Yes, the number of species has been cut in half. If that reduced species diversity would mean that the bay is still covered in coral, then maybe that wouldn't be too bad. I have a second part to that answer, but let me quickly point out one thing I would say, right, what I tried to show on the slide. Not only did species diversity drop, also coral cover and the size of individual colonies is drastically reduced. And smaller colonies, as you may expect, are also then more susceptible to essentially die, like succumb to a disease infection. A bigger colony, the time it takes for the immune response to kick in and maybe fight off that disease, you may only see some necrotic tissue that then quickly grows back. But if the colony is small, and we see that a lot, it will die very quickly. The other thing is, changes in species diversity and composition may have these trickle-down effect, and this comes to the fish. There's another good example in Guam. Coral bleaching has led to mass die-off of staghorn corals in our shallow waters. And if you're familiar with staghorn corals, they form these like very complex three-dimensional structures. When you swim on that reef, you see a ton of fish. They don't seem to be very resilient to these bleaching events, but we have, if you're familiar with coral, Pavona and agaricid coral. It's more like an encrusting coral. That one seems to be thriving, and we take it into the lab, and even under very hot conditions, its polyps are out, it's very happy. So you could say, this is fine, they're growing there, but they support a very different fish fauna, right? So you have a very, I don't know if I like the term, but the ecosystem services that are provided by the reef, right, they change drastically.
Great, thank you.
No problem, thank you.
Thank you, Basti. I'm really curious about the restoration work and the connection to your, and specifically, I assume that's sort of a nonprofit organization, but the government support for sort of addressing this problem and how that's going.
Oh, you mean, so like
sort of management.
Yeah.
More official.
So Guam it feels like a very special place in that sense. So our local government is very interested in these issues. The governor will show up and give an address, right? We just had a conference on island sustainability, and governors, senators, they are directly engaging with this community, and this initiative that I pointed out, they're now also getting funding through Guam Green Growth, and there's some direct government funding for that initiative. And it seems I'm not directly involved with this, the director of our Sea Grant program and the Center for Island Sustainability, he has been spreading the word, and there's now a Northern Marianas Green Growth, and I don't think we can take full credit in Guam for green growth. I believe it started in Hawaii, but there's sort of like the island nations, the islands are talking to each other and recognizing these problems, and legislatures are willing to actually fund and support these initiatives, which I think is fantastic. And there's a connection, right? It's like coral restoration is what do you call it? A lot of people know about this, but there's no point in even trying to restore the reef if there's a constant impact like sedimentation, for example, right? So we have to think about all of those sides of the equation. Thank you.
Please join me in thanking Bastian. And when Bastian was a Buck Fellow, Alan Collins, our last question asker, was his mentor. And I know Alan's very proud because I saw him this morning.